So, now what are the different scores, what are the various parameters used to assess the score? What is the maximum score? This as I discussed earlier, this is the high scoring pairs, how many high scoring pairs in the alignment, right. This is the bit score of high scoring pairs. Then the total score, this will give you the all the HSPs from the same database sequence. Then the query coverage, as we discussed now, the percentage of length coverage for the query, right. If you have 100 residues and the alignment you obtain only for 60 residues, then 40% you do not cover, right. In this case the coverage is only 60%. Then E value, what is E value? This expected number of chances, right, randomly you can get with the same alignment. Why the query coverage is very important?
Sometimes partial match can be used.
Yeah, sometimes you get partial match, right, if you have 100 residues, right. If you use all the 100 residues, you have another match with the 100 residues, then your score will be less. Number of similar residues or identical residues may be 40 or 50. On the other hand, if you have some only some segments, for example 10 residues or 20 residues, and this you will have high matching, for example you have the matching of about 90%, right. Now there is a bias, if you have this one, the random choice to get this specific residues is very high, right, compared with the complete alignment. This in this case you have to take into consideration not the score, but also the query coverage. This is the reason why they give the query coverage also in the alignment score. So, now align the two sequences, this is your query sequence and this is subject 1. So, this is the first sequence and second sequence. What is the middle? If both the residues are the same, then we put the same letter, right, M, K, A and so on, and one place you put plus. What is the meaning of plus? Similarity, right, similarity from the matrix you use, either the PAM matrix or blossom matrix. So, here I and M they are similar, right, they have the positive score, so they put positives. So, based on that they had calculated identities, that means out of 148, right, the 148 sequence, right, completely everything is aligned. So, gap is 0%. So, in this case query coverage is 100%, right. So, out of 148, 147 are matching, right, the identity means exactly the same. So, this is 99%. And then if you see the positives, it contains identities plus the plus, that means similar residues. So, add everything out of 148, all the 148 are positives. So, this is 100%, right, they give along with the scores as well as the E values. So, now here if you give two sequences, right, the earlier one we give one sequence, we try to find the match, all the matches, and then see which one has the best match, that will be similar to your query sequence. Now, if you have two sequences, so I gave sequence number 1, right, and the sequence number 2, and how far these two sequences are similar. In this case BLAST can handle the situation, enter the first sequence, right, in the first place, and here you have to click on this align two sequences. If you align two sequences, then the next box will open. So, in the next box if you give a second sequence, right, and then BLAST, if you do this, right, you get this is the query 1 and this is the subject 1, right. So, you have the alignment here, right. So, there are 115 residues, among 115 44 are same, and among this 115 67 are positives. If you compare this alignment and the previous alignment, what is the difference, right, the previous one it is 100, identity is 99% and the positives are 100%, almost you can see the alignment. So, you cannot say that for all sequences you get this type of alignment. So, if you see here lot of gaps, right, the residues are totally different, you can see Q and V, right, they are not, they have negative score in the blossom matrix or the PAM matrix. So, here there are gaps. So, some cases if it is same they put the same residue, and the plus means they are positives in the case of the blossom or PAM matrix, right. So, now we here see there are 3% gaps, right, and 58% positives and 38% identities, right. So, be sure always the positives are higher or lower than identity, higher than the identities, because positives include the identities plus the similar ones. So, now in the previous class I showed this two sequences as an example to get a dot matrix. If you want to have a dot matrix, here you have the dot matrix view, right. So, if you click on this, then you will get the dot matrix for these two sequences. Okay, we did the dot matrix and we found some matches. How far they are similar? How many percentage this human and chicken hemoglobin are similar? So, what to do? Do blast. So, here if you give the first human sequence and the chicken sequence, right. So, now you give the alignment, how much, what is the identity? 69% identity and the positives are 82%, they are completely aligned without gaps, they are aligned, right. So, this is the sequence identity between human and chicken protein sequences. So, we discussed BLAST and we have another program called FASTA, I discussed earlier. So, FASTA also can do this, if you give the sequence here, right, this is the FASTA sequence, and if you run, then this will give you the sequences and it will give the alignment, right. We can give the values similar to the BLAST. So, till now we discussed about two aspects. One is if you have query sequence, you can get similar sequences in the databases, and the second one pair based alignment. If you have pairs, you can align. If you have more number of sequences, hundreds of sequences or thousands of sequences, then how to make the alignment, right. So, in this case, if we do the multiple sequence alignment, right, name itself tells this is the multiple sequence. We have more than 2 sequences together and you can see whether there is any residues which are similar in different sequences. So, the sequence alignment of 3 or more sequences, right, generally proteins or DNA or RNA whatever. So, what will it provide, which information we get from multiple sequence alignment? Conservation, right. So, these sequences whether they have any evolution relationship, right. So, you see the input of query sequences, we assume that these sequences are similar and where they are similar and where they are distant, right. For example, just we showed example like chicken and human hemoglobin, right. What is the sequence identity? 62%, right. So, if you have different organisms and if you align these sequences, then you can see which region are similar, which region we have maintained the same residues and where we have the variations, and variations happens in which type of substitutions. So, then we can see whether they have a share a lineage or they have any common ancestor, right. You can see the relationship if you align different sequences using multiple sequence alignment. Okay, how to do this, right. This is a 2D table, right, because we give the sequences one by one, right, and put the same sequences in different same residues positions in different sequences in the same place. So, here first we see the relative position of residues, if any residue is preserved. For example, some residues in the PAM matrix we have high score for some residues, right. Can we list few residues which have high score in the PAM matrix? Cysteine, tryptophan, right. These residues are very conserved residues, right. They really prefer to be not changed. So, if you change it may have adverse effects. Likewise, if you see the sequences and any positions or any set of regions where they preserve to be the same in different sequences, then we have similar residues in all sequences, they are placed one by each other, so that they can see vertically the same residue. This is what they want to maintain when doing the multiple sequence alignment. For example, here I give 5 sequences 1, 2, 3, 4 and 5. If you see the different positions, right, I give 10 positions. If you consider the first position, right, so it is accommodated with which residues?
Tyrosine and okay.
Mainly Y and F.
Aromatic residue.
Right, fine. So, the 2 residues. So, they put small y because it is mainly dominated by Y and 2 other residues. If you do a second one, so mainly with the D, so they put small d, and the fourth position if you see G is maintained because G is the same in all the sequences, they put G here, and some cases if you see the 10th one, this is small l, right, because mainly L and V, and some cases they put the equal preferences, right. For example, if you see position number 5 or 6, they put either A or I or either V or L, right. So, in this case you can see the positions, some residues are maintained. So, they like to be the same residue at different positions, right. For example, number 4 the glycine is maintained, likewise position number 9 this alanine is maintained. So, this will help you to see the importance of some residues which are of functional importance or structural importance. So, what is the computational complexity? Because if you align 2 sequences, it takes some time, which depends upon the length of the sequences, right. So, if it depend on the length of the sequence, it will depend on this size for 2 sequences. If you increase the sequences, the complexity will automatically increase, right, because the processing time and the memory space, this will related to the product of the length of the sequences. If you increase more number of sequences, it will take time to align all the sequences and make it a consensus one. So, we extend this comparison, right, pairwise comparison to 3 sequence, 4 sequence, 5 sequence, right, complexity increases. If there is n sequences, then the complexity is very high, right. So, in this case the time taken to compute the alignment is also very high. Then how to do this? How to tackle the situation, right. There are several approaches to tackle the situation. Cluster is one of the most popular approaches, right, to align the similar sequences which are evolution related. So, how it works? First they try to sequence in pairs, right, then similar sequences or small sequences they put together, right, then they form the cluster of sequences which are similar to each other and they put one by one each other, and the distantly related sequences they do it later. Likewise, they can see the correspondence between similar sequences and distant related sequences. So, first what they do, similar sequences are aligned first and the distant related sequences they add later. Once the pairwise sequences are calculated, they use the sequence into groups and then from that they make the final sequence alignment. There are different ways to construct this multiple sequence alignment. So, let us run the cluster W, this is a program which uses the positioning of the gaps also in the close related sequences compared with the more distant ones. So, the information we obtain in the alignment process about the variability of the similar sequences, which will help to introduce gap penalties, because if you have similar sequences, right, then you have less gap penalties compared with the one which are having the distant each other, right, from this query sequence. So, now this is a program cluster W, right, currently we are using cluster omega, right, to align sequences with the several organisms, multiple sequence alignment. How to do this? Okay, we go to the cluster W. So, here you enter the sequence, right, of this multiple sequence alignment, okay, give one by one, right. So, if you give all the sequences in any format, because they accept several formats, right, then you run the program. So, then we get the data. So, from this figure we can see how different sequences are aligned. So, if you see this one, this is the sequence, different sequences we gave, and the next one is the alignment, how they are properly aligned, and then see the residue position in this each sequence. So, if you look into this alignment, we can infer some information, right. What are the information you can obtain from these?
Conserved regions.
Right. First we can see the different color codes, right. What is the meaning of different color codes? What is the meaning of blue color here?
Charge.
Yeah, charge, negatively
Charge, right. Blue is D and E,
negatively charged.
right. There aspartic acid, glutamic acid, negative charge. If you have the green one,
Hydrophilic.
N, S, Y, right.
Polar.
So, there are polar residues. They listed Y as a polar residue because it contains
OH.
OH group, right. So, this is aromatic, but even it contains OH group, and hence the cluster W consider this as a polar. So, they give green color for that. Then what is the magenta one?
Positive charge.
K, here is R. So, positive charge. Then we have the red one, mainly the hydrophobic residues, right, valine, isoleucine, phenylalanine and so on. So, if you see the colors, you can see the same colors are aligned the same position. For example, if you use the E, aligned up to from here to here. So, likewise if you see this position hydrophobic residue, the valine, leucine, isoleucine, so it is aligned for several sequences. So, if you do the multiple sequence alignment, you can see that any specific residue positions are maintained in different organisms. Here interestingly I give the sequence that is mainly the membrane proteins. There are two different types of membrane proteins. I used the beta barrel membrane proteins, I tried to align. So, even these proteins they maintain some conservation, this is why they are having some specific functions for this type of proteins, right. So, you can see the residues are conserved. So, you can see the structure function relationship in any types of proteins. So, now I have a question, how to get the multiple sequence alignment for hemoglobin A chain from different organisms? How to do this?
So we can get the hemoglobin A chain sequence. We can do blast. From blast we can obtain the
Different.
right.
sequence
First
of
we
the-
get the sequence of hemoglobin A. Where shall we get the hemoglobin A chain sequence? Uniprot, right. First you go to Uniprot, right. You check with the hemoglobin A, you will get list of sequences, then you get list of sequences. Then what to do? You have to identify relevant entries. The question is you have to get the alignment for different organisms, but if you do Uniprot, you search with the hemoglobin A, you will get several entries from same organisms as well as different organisms. What is the possibility of having same organisms? Why we get same organisms different data?
mutation data.
Mutation data, right. So in this case you will get the same human, you will get 2-3 times, but the question is to get from different organisms. Then you have to select relevant entries. If you can select relevant entries and then you can save in FASTA format, that we discussed in the one of the previous classes, right, how to obtain the data using Uniprot database. So you can get the sequence in FASTA format and you give these sequences in cluster W. If you do this, right, for take the sequences, now you give this in this place, you can paste sequences and you will get the multiple sequence alignment. So this is we discussed about the cluster W. There are several other software which are available in the literature which can also be used for the multiple sequence alignment. So for example MAFT, right, this is available in the this website cbrc.jp, right, and this muscle, this is also one of the widely used softwares. So you can see this is in UK, so you can use this muscle, and the Promols, this is another software, this also you can use for multiple sequence alignment. So why do we need different software to use a multiple sequence alignment? So what do you expect? You get similar results or different results?
Slightly different.
Yeah, almost you will get similar results, but they try to improve the alignment values mainly to maintain the conserved positions, and second option is the speed, right. We need to get the data very quickly, then to handling more number of sequences and handling the sequence at different lengths. So there are several complexities in multiple sequence alignment, so they developed different algorithms, right, to handle these situations. So you can use any of the software to align the multiple sequences. So in the last few minutes I discussed about SciBlast, right. So what is SciBlast? It's a position specific iterative blast, right. It's a program which searches the database of sequences similar to query sequence, then they get the blast sequences, right, and then we try to derive the position specific scoring matrix. Based on the alignment you can compare the positions and the residues in different positions, then they try to align these positions and derive the position specific scoring matrix. For example, if you have your own sequence, what is the possibility of having the same residues at different positions depending based on the aligned sequences in your homologous sequences. How to do this? First is search the results with the blast and derive the patterns from the multiple sequence alignment from the initial kits. Then they repeat the process, right, because for example if you have two alignments, right, they are very perfectly matching and the third one is matching with another for example 10th position and 11th position, they iteratively they change and see if there is fine tunes with the best match, then keep at the top, then again the rest they tune again. So during iterative process finally they make the perfect alignment. This is how the SciBlast works. Once they make these alignments, then they try to see what is the probability of having a specific amino acid residue in a protein to maintain in the different organisms of same type, right. So this is the site you can use, the SciBlast. So here we give your input sequence. Once you give the input sequence, what it will do? It will do the blast, it will collect the sequences and do the multiple sequence alignment, do different iterations. You can see the threshold value. So depending upon the expectation value, so iterate again and again, and finally if you click here, then you will see your alignment, multiple sequence alignment, plus you will get these position specific scoring matrices. Now these position specific scoring matrices you can use it for further applications. For example, if you have a sequence of unknown function, right, and if you want to see which residues are binding with some other molecules, right. So what can you do? You will get a sequence, right, then you can see the similar sequences and then we can make this matrix and see where it matches with the known sequence, and mapping the known sequence with your own sequence you can see this region could be a probable binding site for your particular region. Likewise, you can have several potential applications by using these SciBlast profiles and the development of these position specific scoring matrices. So, by summarizing, what did we discuss today?
BLAST, different kinds of algorithm, for example BLAST, multiple sequence alignment,
Yes,
and then SciBlast. So these
Faster cluster.
cluster W, right. We can started with the pairwise sequence alignment, right, the two different algorithms like the BLAST and FASTA. Then we extended with the multiple sequence alignment and see what are the various parameters you can use to get the score. How about the sequence identity, what about the sequence similarities, right, and how far the two sequences are similar or different from each other. Then we extend to multiple sequence alignment. Then using the multiple sequence alignment, you can see the position where we have the similar residues maintained in different organisms, and then you can also use these alignments for deriving these position specific scoring matrices, that I will explain in later classes. So, in the next upcoming classes, I will discuss about the use of this multiple sequence alignment to get the conservation score, how to identify which residues are conserved or any metrics to get the numbers using some numerical values, or you can have this alignment based on any trees, whether you can relate form of these evolutionary trees, right, as well as what can we do with these specific alignments and so on. Thanks for your kind attention.